Hi everybody, this is Dr. Van. I'm back after I've finished my 2 plates of cell or seeding the chemicals on the cells for cytotoxicity assay. Now we have to prepare our neutral red solution for what we're going to do. And so I'm just using the paper that I provided for you guys. That our neutral red came from Sigma at 3.3 grams per liter, which is 3.3 milligrams per liter. And so the paper said we need to add it at 40 micrograms per milliliter. So basically we need to dilute it by a certain amount. I'm actually going to let you guys do the math. And so to do this, I'm going to get a known amount. I think there's 24, so that they suggest using 12 milliliters per plate. So I think I can use 24 mLs of my media, and then I'm going to put the appropriate amount of neutral red in there to go from 3.3 milligrams per mil to 40 micrograms per mil. I just barely have enough. There's some bubbles and maybe even not quite right, but this will work. And make sure it's mixed decently in there. So this stuff we do keep the neutral red sterile. We don't want it getting contaminated either. Just sort of make sure we wash out our media. Wash it out with the media. Yeah, so that's why we're doing this in a biological safety cabinet, to keep it sterile. But now we're done. If you notice from the paper, it also said to store this in the incubator with our cells. And so that's what I'm going to do. I'm going to stick this tube in the incubator with our cells, and then tomorrow when we add it, the protocol also says we need to centrifuge it first, because if there's any crystals in there, we don't want to put the crystals on the cells. And so that's what the first thing I'm going to do tomorrow is separate this into 2 15 mil tubes, 12 mils each, and then centrifuge, and then we'll add it to the cells and let it sit there. And then we'll get a reading of cytotoxicity from the neutral red. So I'll see you guys tomorrow.